If all living organisms are made up primarily of proteins, and what we see when we look at them are the proteins that make them up, protein synthesis becomes a really critical process to understand when it comes to biological systems. So we spend a lot of time in Principles of Biomedical Science helping students understand the importance of this process as well as the nuances of how the process works. So I'm going to use a paper model to demonstrate some of the different concepts that go along with this, and then I will include the resources at the end in case you would want to use these with. So the first part of protein synthesis happens inside the nucleus, and inside the nucleus we would have the RNA polymerase, which is represented with this blue piece here. We would have the DNA, double-stranded DNA, that would also be copied to make the protein that we're trying to create. So this would be the coding for that on the DNA template strand. And then we have this, which is going to serve as our mRNA strand. So the first part is called transcription. And during transcription, RNA polymerase comes into the DNA and unwinds it, so it is single-stranded. And then basically we use the template strand, and RNA polymerase will use nucleotides to help create a complement to the template strand. So a complement means that the opposite base is used. And in the case of mRNA, which is in the green, there is no thymine. So thymine is substituted with uracil. So you can see that an adenine would match up with uracil, and then the thymine would code for an adenine. And so you could just kind of follow this all the way along the whole gene of interest. All right, so this is all happening inside the nucleus, and we would basically have the strand kind of complete and move through with the RNA polymerase protein helping facilitate that whole process. All right, so this all happens in the nucleus. Now, the mRNA that was formed inside the nucleus through the process of transcription will move out into the cytoplasm of the cell. And here, it will work with ribosomes and tRNA to help produce the protein. So it'll make an amino acid chain. So basically, this is floating around until it meets up with a ribosome. And the ribosome will read 3 of the letters at a time. And it will keep searching those 3 letters at a time. These are called codons. It will search until it finds a start codon. So our very first start codon is right here at AUG. Now, to figure out what amino acid that codes for, you need a codon chart. And so here's an example of what a codon chart would look like. And basically we have the AUG codon. So you start with the A, you move to the U and the G, which is methionine, which is a start codon. Okay? And so that would be the first amino acid that would start building in our chain. Then the ribosome shifts over to GCG. So you go to GCG, which codes for an A or alanine. And this is all being brought, the tRNA has an anticodon, which is the opposite of this, and will actually bring the amino acid in and help form the peptide bond between the amino acids. And so you'd keep going in this way until the entire protein was built, right? So you have CCC, which stands for the P or proline, and so you keep building along as you go. In the end, you would have a completed amino acid sequence, and then with that completed amino acid sequence, there would be some folding that would happen to finalize and finish the protein. But all of this is protein synthesis, and these would form all of the different proteins in your body, from enzymes that help you digest things to the RNA polymerase that helped unzip the DNA in the transcription process. So super critical process, really useful for students to understand biological systems. I will include in the last slide of this video the place where you can reach these resources if you'd like to use them, these paper cutout resources if you'd like to use them with your students.